Or me, okay. Hi, I'm Rick Rachubinski, and I'm professor in the Department of Cell Biology at the University of Alberta.
Andrew Simmonds, I'm an associate professor in the Department of Cell Biology at the University of Alberta.
Fred Mast, I'm a graduate student in the lab of Richard Rachubinski at the U of A.
Okay, so tell me about the discovery that was just made in both these labs.
Well, the discovery that was made in, as you said, in collaboration with Andrew's lab and my lab, was to look at developing a fly model, a simple model organism of a human genetic disorder called Zellweger syndrome. And this really was an important development because though other people had tried to develop model systems in more complex organisms such as mouse, the inability for those mutant organisms to come to term was a real detriment to further studying the Zellweger syndrome. So these diseases are very severe human diseases in which the individuals afflicted failed to develop an organelle called the peroxisome, and therefore they have a lot of metabolic disorders that are associated with them and die soon in their infancy. But the Drosophila model, the fly model that we've developed, allows us to study this disease in depth. Also will allow us in the future to be able to look at drug molecules as therapeutic treatments, potential future therapeutic treatments for humans. So it's a substantial development and one that we're very, very excited about.
Okay, and why don't you guys talk about the next step in the research now?
We'll let Andrew talk about that.
Well, one of the sort of the big advantages of working in a department like cell biology is the availability for collaborative research. Our lab has modeled several diseases now using Drosophila, and working with Rick, the Zellweger's model is one more. And the advantage to flies is they're very, very small and they're very, very easy to work with. So like Rick alluded to, the drug studies we can do rapidly and quickly, basically looking for an improvement in the flies to see if they lessen the symptoms. And what's nice is these are the same symptoms that are analogous to what we see in human patients. So the idea is we screen a lot of drugs quickly in the flies because they're small and cheap, and then that gives us a leg up. We can then move to working with human patients, and we've actually identified other collaborators across Canada who are quite willing and eager to work with us, who have several human patients to sort of do the next step for these drug studies.
Okay, and Fred, just talk a little bit about the importance of this discovery and the research ongoing from here.
Sure. Well, I found it to be quite an exciting study. One of the key parts of that was that we did this genome-wide analysis of transcription when you have compromised peroxisome biogenesis. And this really allowed us to get a kind of a complete picture of what's going on in a multi-cellular organism. We saw downregulation of a lot of genes involved with cell growth. We saw upregulation of genes involved in innate immunity, and I think this going forward will be a useful tool to start to find new drug targets potentially, but also just to monitor as we move forward with future studies just the health of these organisms.
Okay, thank you very much, guys.
Thank you.